I like Devil's Villain. I think it's better than
No,
he's gonna murder people and just attack other things.
I mean that is true.
I mean, he did used to do like they changed designs. He's like thinner. Yeah, see, very much. Like,
All right. So we're gonna start with leishmaniasis. So we've got an overview here. We've got the parasite here in a histological stain. We've got its vector in the top right corner, a sandfly. And then we've got a couple of different types of leishmaniasis here. So, in terms of species for Leishmania, so it's color coded here, and let's just remove this, put it over here. It's color coded to show you the ones that are capable of, causing like mucocutaneous leishmaniasis, cutaneous or visceral. And we see that red are considered the New World species. So the New World being on the Western Hemisphere, Old World being the Eastern Hemisphere. And we see here that there are a lot of New World species highlighted here in red. And of these, thankfully, only a handful of them are capable of causing mucocutaneous leishmaniasis. And that is this form here. So it's a very disfiguring form. Cutaneous, that's this bottom right-hand one. And then visceral, we'll talk about it. It's more of like an internal disorder than like an external one. Okay. And one of the issues that we're going to see is that for this parasite, many of them are zoonotic. Has anyone heard of zoonotic before? If not, you've likely heard. So yeah.
Is it that they
Yeah, so they can spread between different species. So certain diseases out there are considered zoonotic or zoonoses because they can infect different species. And like COVID-19, for instance, that's a zoonotic disease, excuse me, that spilled over from another mammal to us. Doesn't necessarily have to be a mammal, though. Yes.
What do the V and the L's mean?
So that is meant to tell you the type of leishmaniasis that it causes. So these are going to be visceral. I'm blanking on the L at the moment because they're not showing C on here.
Why are there visceral leishmaniasis in the cutaneous column?
So they've actually observed that originally they were visceral, but then they observed, hey, they can also cause mucocutaneous. So it's not so important, the V's and the L's in this case. The takeaway from this is a lot of these can cause cutaneous, very few, thankfully, can cause mucocutaneous, and even less can cause visceral. So, as we've seen in the previous slide, here, and on this slide, the disease, leishmaniasis, is globally widespread, but the diseases will vary. And so, as mentioned in the previous slide, the Old World, we define it as Europe, Asia, and Africa, New World being the Americas, okay? And we're going to see that visceral leishmaniasis is mostly going to be Old World. We do see some of it spreading into the New World. We're seeing cutaneous a lot of it being in the old world as well. And now it's starting to spread into the new world. And where we're seeing mucocutaneous, it seems to be mostly isolated to the new world at this point when the data were collected. Doesn't mean it can't move to another sector of the world. It's just that version has to, get globalized, accidentally get transported there. Okay. So it is another kinetoplastid. So same thing as last time when we were talking about the trypanosomes. We've got the same morphological forms, the different types of trypomastigodes, epimastigote, etc. Again, just like with the previous kinetoplastids we talked about with trypanosomes, they've got two types of extracellular DNA, or extranuclear DNA, I should say. So all of this is the same. Okay? So this slide is also the same. Remember, we were talking about their kinetoplastids because they have this elongated mitochondrial structure called, and within it, we've got this thing called the kinetoplast. And the kinetoplast is a unique set of DNA arranged in in both maxi circles and mini circles. And this set of DNA is essential in RNA editing. As mentioned, it'll cause various diseases, but we're really going to be focusing in on the human diseases. But there are associated animal diseases, but that's not the focus of this course. So, with that said, we've talked about where do we. Yes, sir.
Could you say what the kinetoplast actually is?
So the kinetoplast is a structure within this elongated mitochondria, and that's where we find the maxi circle DNA and the mini circle DNA. So with that said, we're going to go a little into detail about the vectors for this, and we've got two different groups, two different species of sandflies. So we've got Leishmania and we've got Phlebotomus over here. And as we can see, at least with what's currently known, there's over 650 plus species of these vectors out there. So that kind of explains why we see such, a massive distribution across the world. Kind of like what we saw with malaria. There's just tons of vectors out there. Same thing for Chagas. So vector control going to be a bit difficult on that. And down at the very bottom, this is typically where we happen to see them. So the phlebotomous species, those tend to be old world. And then new world happens to be the Leishmania species. That's just where we tend to find them. Okay, so these next few slides are going to be busy as usual because we're getting into the life cycles. Okay? So, we're going to, excuse me. We're going to kind of fill in the left-hand side first, go through the blanks, and then we'll put all of these words to the images after that, in this case. Okay, so, like what we saw with, previous kinetoplastids, there's a lot of different physiological forms in, the hosts. And we saw with, the trypanosomes in the sandflies that, there are a lot of different forms within the fly. So that's what we're going to see here as well. Okay, so there's many different forms within the female sandfly. Again, we tend to see female flies of these different species being the ones that do the blood, the eating of the blood. The male flies, they'll eat other things, but they won't bite humans. It's like they'll go and get like pollen and things like that. So, female sandfly is going to take a blood meal. Thankfully, in this case, it's not as painful as like a horsefly or, the others that we've talked about, like the tsetse fly. So it's going to take a blood meal, okay, of an infected host. And what we're going to see, and this will correspond to number 1, and we'll get to it, is that excuse me. The amastigotes that they're taking up are going to, so here for this first part, the amastigotes that they took up within the blood, they're going to differentiate into the procyclic form of the promastigote. So we're going amastigote differentiates into a procyclic promastigote. And so we got this chart here on the bottom to kind of help you track this as well. Okay, so we're at this bottom part here. Amastigotes are being taken up. They'll differentiate into procyclic promastigotes inside this female sandfly. And then we're going to see that replication, okay? And the procyclic promastigotes, excuse me, are going to do replication via, what do you think? Binary fission as usual. So procyclic promastigotes replicate via binary fission, and then they're going to differentiate into this next form of a promastigote outlined down here, called a nectomonad. And once they become this procyclic, wow, so many terms of blanking. Procyclic, sorry. Wow, I'm messing them all up. Nectomonad promastigote. Once they become that nectomonad, they're actually going to physically attach themselves to the midgut region, okay? And one of two different things can happen here. So we're going to focus on the first part for 3A. So these nectomonads, they physically attach to that midgut, and they will differentiate into this leptomonad. So right here, they differentiated into a leptomonad form of a promastigote, okay? But they could also, become a metacyclic promastigote. So they've got two different options here. So what we're going to see at this point is the nectomonads have become leptomonads, and the leptomonads can go into one of these two forms. And they're both promastigotes. They can either become a haptomonad or they can become a metacyclic promastigote. So we're at this point here in the little bottom chart where we're seeing haptomonad promastigotes or metacyclic promastigotes. Moving on to 3B, they're able to maintain the infection within the female sandfly for the life of the host. So once that sandfly is infected, it's infected for life. And so it can continue to, take blood meals and then infect more hosts at that point. What we're going to see at this point, though, is say the leptomonads differentiate into the metacyclic chromastigote down here. These metacyclics can be released either through, a blood meal, okay? So that's how they can be released, or they can turn into this other form called a retroleptomonad. And then depending on the environmental signals within that sandfly, it can either continue to just stay as this retroleptomonad, or it can differentiate into the metacyclic chromastigote. So it's like this part right here, these last three kind of arrows, like it's a metacyclic or it's a retroleptomonad. And so it can just keep switching between these forms. So the answer to 4 is that for the metacyclics to get released when they're taking a blood meal, the host that is going to get released via the blood meal through the salivary glands, or it'll just revert and turn into this alternate form called the retroleptomonad. And what that's going to do is it's thought to just increase the parasite burden inside the host. So it's like you've got this stable, steady state of an infected parasite in the host, and it can just keep making more of itself.
So it just.
Right. And so then depending on the signals within it, it's like, all right, time to switch over to metacyclics, or hey,
It can activate itself.
yeah. Yeah, essentially. So it's like you've always just got this infective state in there, and then it's like, all right, time to go and infect more vertebrates. Let's switch over. So I know that was a lot and that was confusing. So let's just kind of go through the image down here on the right-hand side. So my suggestion for you when studying this is to use this bottom panel where it's showing the progression of the forms while you're looking at this right-hand image, okay? And in the very center of this, so we've got this whole view of the female sandfly showing you the locations of these different forms of the Leishmania parasite. And then all of these zoomed in insets are corresponding to the different regions in here. Okay, so that's to orient you on this very complex image. So it's like we've got one here kind of towards the end, well, towards the bottom of the abdominal midgut. That's this right here. Okay, so we've got a blood meal. They've taken up the amastigotes. Okay. These amastigotes, as they're migrating from the mouth, getting towards the back end of the abdominal midgut, they're going to differentiate into these procyclic chromastigotes down here. Okay? And then what we're going to see is that those procyclics, they're going to move up a little bit in the abdominal midgut, and they're going to differentiate into this next form called the nectomonads. All right? So we're still here. We've moved a bit anterior within the abdominal midgut. And then we've got 3A and 3B. So as we're transitioning from 2 to 3A, we've got those nectomonads, and they're going to escape from this region called the peritrophic matrix. And now we're seeing that they're going to physically attach to the midgut. And the explanation is down here as to why they're doing it. They don't want to get expelled via the hindgut. It's like they're so close to essentially the rectum. So if they don't physically attach, they'll get flushed away, and they can't continue the rest of this life cycle. So we've got the nectomonads, they've got a little flagellum, they've now physically attached to the midgut, and they're just going to physically differentiate, so they're going to transform, bless you, and now they're going to turn into this leptomonad form, okay? And we see that they're physically attached. And the way that they're, they're kind of sticking in here is they've got this little gel they've got on. What we can see at this point is the leptomonads have two choices. They can either turn into the metacyclic form over here at 4, okay? Or they can turn into this haptomonad form. So here's 3B. Let's say the leptomonads are like, no, we're going to turn into this haptomonad form over here. And that's happening towards like the thoracic midgut to foregut. So it's like they've migrated and they're just kind of staying here.
Okay, yeah, so the heptobone has a dormant.
It's like one of their kind of dormant-ish phases, if you want to think of it that way. It's like they just moved further up the midgut region. They've gone from the abdomen to the thoracic midgut foregut region. So, say they go this metacyclic route. Well, we're here at 4, so again, they've been kind of like crawling up at 3A. They've just been kind of crawling their way up to get over here towards the head region. So they're in like the thoracic midgut at this point. Okay. And it's at this point that the leptomonads can differentiate into that metacyclic form. And this metacyclic form is the infective form for vertebrates. Okay? And so they'll just migrate, they'll make it into the salivary glands, and then when you have a blood meal, it gets transmitted. Or they can, turn into this retroleptomonad and just, further kind of stay there and then just increase the burden. So takeaway from this, there's a lot of complexity within, one of the hosts. I'm sorry?
What happens in the other hosts once it is infected?
So we'll start to get into that.
Uh,
Yes.
So when the haptomonad, it differentiates into that. But the metacyclic, they're both in the thoracic area. It's just the haptomonad stays there.
Yeah. We're gonna just kind of leave it at that because like it can get far more complex, and I'm just like, here's part of the life cycle. We're just gonna put that off to the side. And so that's a big problem with parasitology is like we're still discovering so much and it's like, what is actually going on in here? Okay, so we're going to transition away from, what's happening within the sandflies, and we're going to now move on to, well, what happens when we've got transmission into a human at this point, okay? So how do the human hosts, emphasis on try to combat this thing, okay? So we've got our innate immune response. We've got our adaptive immune response. They need to talk to each other to have an effective immune response for most things. So, excuse me, that's not too novel here. So, in terms of some phagocytic cells, what are some phagocytic cells that you might remember? Okay, macrophages. Yeah, neutrophils. There's another. Dendritic cells. Again, you typically don't think of dendritic cells as a phagocytic cell, but they do because they have to, absorb or take in stuff so they can then go present it. So, phagocytic cells, cells that just go around and engulf something, bring it in, break it down. So we've got our macrophages, our neutrophils, our dendritic cells. Okay? We've got ROS and RNS. We've talked about these briefly before, but just to, prime you again, it stands for reactive oxygen species and reactive nitrogen species. And these are actually very potent forms of chemicals in your innate immune response. They're actually super excellent at like killing things. So they essentially will just bind to something, and because of chemical modifications that happen that we won't go into detail on, they inactivate it. So we'll just oversimplify it. ROS and RNS bind to things, alter the chemical structure, and inactivate it. So that's why they're really good. And that's why if you don't keep them under control, it can be really damaging to us or, other hosts. Mast cells. So that's another example. We won't go into, much more detail than that. Was like mast cells exist. We'll leave it there. The complement system. We've got 3 different types of it. We're going to just mention the names. So we've got the classic pathway. We've got the alternative pathway. And then we've got the lectin pathway. For these 2 blanks down here, we've got different types of proteins. So we've got things called cytokines, and then we've got chemokines. And then finally, at least of what we're gonna kind of briefly talk about, we've got these things called PRRs or pattern recognition receptors. And there are so many different types of them. There's toll-like receptors, there's nod-like receptors, there's so many others. Okay? But the reason we've highlighted the toll-like receptors here is because that's going to be like the main focus here. Okay, I'm not saying the others aren't important, but toll-like receptors, there's so many different types, and they're really important for kickstarting a lot of stuff. Okay. Transitioning over to the right-hand side for the adaptive response, we've got different types of helper T cells. So when we talk about T helper cells or helper T cells, we'll abbreviate them as TH and then whatever number. that corresponds to it. So this would be T helper 1, and this would be T helper 2. Those are some of the main ones we typically talk about in parasitology and immunology, and even virology. There are many other subtypes of helper T cells. Then we've got our natural killer cells here, so NK. But there's a major player that's missing here. What would that be for the adaptive side? Yes. Yes, that is correct. B cells. So, question for y'all to think on for like 30 seconds or so. Why do you think we need both the innate and the adaptive to have an effective immune response? So even if you know right now, we're just going to let you think on it for a moment. Yes.
So, is the fact that B cell is not an adaptive immunity, is that implying that B cells are unrelated to any response?
No, I just wanted to have an interactive question.
Oh, okay.
Yeah. Bless you. Okay, why do we think we need both arms of this immunity to have an effective immune response? Yes.
If I remember very basically,
Mhm.
B cells, they have to be shown what the pathogen looks like. And then I think they kind of tell the phagocytic cells what they should be looking for.
Partly, so they definitely need to be told what is out there. So they're going to need, and this is where we're going with this. So we need the innate side, like dendritic cells, to go over here and talk to the adaptive side and be like, hey, T cells, do you recognize this? And they're like, yes, we do. All right. Now go and talk to the B cells and tell the correct B cells, hey, we found this, start making antibodies. And then those antibodies can then feed back into like opsonization and phagocytosis. It can also lead to inactivation or destruction. So we really need both parts of this to talk to each other to be effective. So that's why if an individual has like a defect somewhere in the innate side or the adaptive side, you'll start to see certain diseases pop up in those individuals that you normally wouldn't in an in quotes healthy individual. And that's just because like part of the system is failing. Okay, so here is how Leishmania can be recognized and how it can get around it. So the overall picture here, excuse me, is just again to show you how the host, so the human, and the pathogen interact. And I do want you to know that yes, there are going to be some slight differences between the Leishmania diseases and how they interact and how they escape. I'm going to kind of just leave it at that. It's like that'd be like an advanced parasitology topic. One thing we're going to see is, like cutaneous, visceral, and mucocutaneous. We'll talk about those different ones, but the way that they interact and escape is different. So we're just going to kind of try and do a general view here. Okay. So I do want you to know this information here in this left-hand table. So you will need to know T helper 1, T helper 1 cells, T helper 2 cells, the cytokines that induce them to do the things that they need to do, Okay. They've got certain key transcription factors that's going to be important. And then you will need to know the cytokines that they release or secrete. So all of them, not just the ones that are highlighted in red. And then their general response. So we're going to see helper 1 subtype is involved in intracellular pathogens. So they're really dealing with cell-mediated immunity. And T helper 2s, they're going to be doing extracellular work. So they're going to go after the extracellular parasites with the help of antibodies. So I know there's a lot going on here, and we're going to go into it. One thing we're going to see, though, from this whole image, is that weirdly enough, Th2 cells can actually suppress the Th1 response. So I'm going to write that on the board. That is very important. And this is bad. And the reason for this is if the Th2 response suppresses the Th1 response, it can actually lead to disease progression. So what we can see with a lot of pathogens is that they can bias the immune response of the host so that they can escape. They can just basically like, hey, look over that way, and then they just run off. That's essentially what's happening here. You need the Th1 response to control leishmaniasis. Okay, so the reason I've highlighted 2 particular cytokines, this is going to go into how they can mess with the helper cell responses. So Th1 can secrete interferon gamma. And what can happen is elevated levels of interferon gamma can actually suppress the Th2 response. Whereas Th2 secreted interleukin 10 can do the opposite. Interleukin 10 can suppress the helper 1 response. So interferon gamma is antagonistic to the helper 2s, and interleukin 10 is antagonistic to helper 1s. So in terms of what I want you to take away, besides like, hey, it's very complex, we've got these bits of information over here that are in brackets. Please note those. Okay, so things that would lead to control of the disease. Well, we need We need like dendritic cells to present the antigens. We need certain pro-inflammatory cytokines and chemokines to be upregulated. We need, productive antibody production. So we need the correct antibodies to be made, okay? And what that can do is it'll decrease the size of the lesion. So when you saw that one individual with like the bite on like their lip, that's a lesion there. And then, if you have an effective antibody response, you can decrease the parasite load within the humans. So that's good. That'll lead to disease control. And then here we've got ROS and NO. So reactive oxygen species and nitric oxide. So nitric oxide is just one of those reactive nitrogen species. So if you were to just say ROS and RNS production, that's fine. And then here for disease progression, we've decreased antigen presentation, so we can't activate certain T cells, and therefore those T cells can't talk to B cells. What we'll see then is that, without, antigen presentation going on, we're not going to make antibodies. All right. Therefore, lesion size is going to just keep increasing. That can lead to like the eating away of the face. Then we'll see, that's happening because the number of parasites is increasing. And we'll see that reactive oxygen species production is being inhibited. Here we've got two of these proteins. TGF beta, I don't know why I put TNF here. So I'm going to change that. Okay, change that. And interleukin 10. So originally I had it here as TNF beta, but I saw it didn't align with this down here, which was TGF. So I was just trying to make it match the infographic. So key takeaways from this very busy slide. Know the table over here. In particular, definitely know these two red cytokines here and what they do. So interferon gamma suppresses the Th2 response. Interleukin 10 suppresses the Th1 response. And down here we've got, here's generally how they respond. And then just know these bits of information under disease control and disease progression. I'm not so much interested that you know all of this other information in here. This is just to give you an idea of how disease control and progression happens. Okay?
So what do you want to go over there?
Just this part here, so where it's got in brackets disease control and disease progression. All this other stuff in here, it's like you can just kind of ignore that. So as you can see, it's very complicated. This is probably one of the more complicated cases we'll be going into for the semester. So, looking at it a different way when we're talking about the parasite strategies for success, that's going to be this next slide here. And we're going to see that for part one, here we're talking about the sandfly. And, within its saliva, it's going to, have the promastigotes, okay? So sandfly secretions enhance promastigote success through these three mechanisms. So one, altering blood flow at the site of the bite. That shouldn't sound too unfamiliar to you. When we've talked about like an ectoparasite in say 101 or 202, the leech, for instance, it's got like an anticoagulant that it'll release at its bite site. So they can then just cause blood flow to increase and, take up watered down blood essentially so it can get as much as it wants as quickly as possible. So, yes. So we're going to see here this thing called this maxidilin. That is the Leishmania peptide that is being released here. And its whole job is to cause vasodilation. So it's going to increase the size of the arterioles. And so then more blood can flow in there and, it can get in and escape real quick. We've got immunomodulation, so altering the immune response. And what we'll see here is that within the secretions of the saliva, it's going to inhibit activation of two different cell types. So it's going to inhibit the activation of T cells, but it's also going to inhibit activation of macrophages. And so it's targeting two really important players in both the adaptive and the innate immune response right there. And so it's already kind of causing dysregulation. Macrophages. And then, really cool, in my opinion, is it's altering chemotaxis. So it is going to alter how certain cell types get attracted to the wound. So, besides inhibiting activation of macrophages, it's going to inhibit attraction of macrophages. It's going to inhibit attraction of dendritic cells as well. Not inhibit. It's going to promote attraction of macrophages, dendritic cells, and neutrophils. And what we're going to see is that they'll try and, take it in and unfortunately get infected. So like neutrophils here, they'll come into that site and they'll take them up and, if effective, they can eliminate them. But in this case, no. So they'll end up surviving inside that neutrophil and disease progression will happen. For the second part down here, Leishmania's virulence factors, we're going to see that it's going to secrete certain peptides that will cause the sandfly to do the following. It's going to cause the sandfly to take up the maximum amount of blood that it possibly can. Why do you think that is a good strategy for success for the parasite? Yes. Okay. But what is it also potentially taking up? Yeah. So it's going to take up more blood. And so then there's a higher chance for more and more parasites to, get into that sandfly, assuming the host is infected. Yes. So we've got our macrophages, our dendritic cells, and our neutrophils. So it's going to secrete peptides to cause the sandfly to take up as much blood as possible so that more Leishmania can get inside sandfly, increasing, its success rate. we've got maxidilin here. Again, we've talked about, it's altering blood flow, but it's also altering the immune response. So it's going to prevent certain chemicals, certain peptides from doing things. So it's going to prevent TNF alpha release. And it's going to upregulate two different interleukins. It's going to upregulate interleukin 6 and interleukin 10. That was the one, yeah, that was that main one from T helper 2s. Whoops. So, it's promoting interleukin 2 production, which is going to inhibit Th1 cell activation.
So the other one.
Interleukin 6 will be important, but really interleukin 10 is the more important one. Like, I could ask a quiz or exam question, but like, what interleukins is it altering, the profile of? Okay. And finally, again, for number 1, though, for sub bullet 1 right here, it's going to stimulate production of a prostaglandin called E2. So we covered these 3 here. It's preventing TNF alpha release. It's increasing synthesis of interleukin 6 and interleukin 10. It's stimulating the synthesis of a prostaglandin E2. It's also going to decrease interleukin 12 production. So it's definitely altering, cytokine, chemokine production. And that's really how cells talk. And then it's going to produce this thing called CPB, which is a cysteine protease B. That's a really effective protease. But taken together, what we're going to see is that CPB, its job is, unfortunately for us, is going to allow Leishmania to infect macrophages. So the cysteine protease B will enable Leishmania to infect macrophages. And so altering all of these chemicals, whether they're increasing them, decreasing them, taken together, that's going to downregulate Th1 response, and it's going to promote the Th2 response. And what we said over here on the board is that we really need that Th1 response so that we can control disease progression. And so it's altering the immune response profile so that it can't happen, or it can't happen effectively. So there are 4 types of leishmaniasis. We will really discuss 3 of them, though. So the 4th one is just called diffuse leishmaniasis. But for this topic in this course, we're going to talk about cutaneous, mucocutaneous, and visceral. And as we can see with visceral, we've just got these dotted lines in here because it's affecting internal organs more so than the outside of the body. Okay. So with that, we're going to stop here for today. And next class, we'll start with cutaneous.